oxygen to get all of your muscles so that you can either fight or flight. You need oxygen to go to your brain, oxygen to dilate your eyes so that you can take in everything that's going on. And it's going to dilate the arteries that are going to your extremity, and it's going to shunt the arteries that are going to your like your stomach and your digestive system, right? When you're in a rest and digest mode, and you're relaxing, you're watching TV, or you go home and you're sleeping, your parasympathetic nervous system is going to kick on. It's going to shunt the blood from your extremity and point it towards your organs and help with that digestion and stuff like that. So it's kind of like a way that your body is able to move blood to be able to get into the type of tissue that you need to do with what you're working with. And so it's kind of interesting how your body does that, like subconsciously, like your body's controlling where blood's going in terms of what you need. And so when you think of that, when you're stressed out, your blood pressure goes up, all these different things, know that that's your body's natural mechanism to be able to deal with stress. So it's not your stress that's like causing that. So it's like it's a effect that your body does to be able to handle it. So knowing that information, you're walking into the TBL on Wednesday, your heart starts pumping, right? You're getting nervous, you're getting stressed out. That's your body saying, all right, let's fight, let's do this thing. Like your body's getting prepared to take on a challenge. So don't be afraid of stress. Use it to energize you to get through it. So, all right, move on to capillaries. And so, what's nice when we're talking about this, your capillaries have a lower amount of blood pressure, right? So it's about 10 millimeter mercury. It's allowing enough flow so that the red blood cells go through the capillaries, but it's not enough to where it bursts the capillaries. There are three different types of capillaries that we'll talk about. We already mentioned the continuous and fenestrated. There's this other term called sinusoid, and we'll get into that in a minute. Continuous is what you're going to find most commonly. It's going to be out like in your skeletal muscles and stuff like that. Fenestrated will be in a select few locations as well as sinusoid. So your capillaries, like I said before, be responsible for this exchange of nutrients, right? It's the point in which your red blood cells will dump off its oxygen so that it can be picked up by the tissues that's in that area.
Bless you.
So that it can be picked up by the tissues that are in this area to then use that oxygen for energy. Another misconception, I guess, that I had when I was taking undergrad and stuff and doing this was that there are arteries that will go through your entire body, right? There's multiple different directions. So there's arteries that are going to, your stomach. There's arteries that are going to your colon, to your liver. They're like separate arteries that are branched off. And then those separate arteries meet up with other capillaries. So it's not like, I guess this is the way that I thought of it, which was incorrect, was that the arteries go through and it cycles through the entire body, and then it meets at one point, and then it goes into the veins and then cycles back through and stuff like that. So know that there's multiple branches of these arteries and arterioles that are meeting up at each individual organ, each individual like muscle and stuff like that. That's helping circulate this blood, so that it's getting these nutrients to each of those muscle tissues. All right. So this is showing this structure being the capillary. These are all the red blood cells that are lined up back to back to back. So this would be called a what? Yeah, I don't know how to pronounce it.
Stacking.
It's like
Rolex.
Rolex or something.
The Rolex. Yeah, Rolex.
Yeah. All right. So what is? I know that's kind of off. What is this structure right here? Yep, red blood cell. So then what is this? What?
Capillaries?
So it's the nucleus of
Oh.
the endothelial cell that's surrounding. So you can see all this right here. Any guess what this is?
It's a platelet? I don't know.
Yeah, so this is, yep, so there's gonna be tight junctions all along here, right? I don't know if you mentioned that, or if you put that there. No, so that'll be on the next slide here in a second. But this right here, you'll see that there's tight junctions within these capillaries that help keep those red blood cells from being able to go out. We'll talk about the sinusoid capillaries that I mentioned, and those actually have spaces that are big enough where some of the red blood cells can like leak through. But for the most part, with like the continuous capillaries, these tight junctions help keep a lot of the fluid in here, and then it really just allows for like gas exchange and for the nutrients to exchange. So this will be your endothelial cell going around, right? This is your endothelial nucleus, and your red blood cell in the middle. This is giving a really good depiction of the difference between them. So you'll see here we have the continuous capillary at the top. They have these tight junctions, right? Endothelial cells, red blood cells going back to back. So there's not a whole lot of space. Fenestrated, they have these pores that I was talking about, right? And so that's going to allow a little bit more stuff to go through. They're going to be found in the kidney, small intestine, and a couple other places we'll talk about. This is your sinusoids, and there's going to be larger gaps. They will have some tight junctions, but these larger gaps allow more things to come through. This is going to be found in your bone marrow and spleen, and also in your liver. So when we are asking questions on these three, the big takeaway is knowing the difference between the three, and then where you can find them throughout the body. Okay. And that'll be labeled here in a second. So continuous capillaries, like more specifically, these are the most that are common, right? They're abundant in the skin, in the muscles. They're connected by tight junctions, which we saw. They have some gaps, but not as many as the fenestrated, right? So they don't have those pores. There are some gaps called intercellular clefts, which allow the passage of some fluids and small solutes, but not nearly the amount of the fenestrated as the other one. So this one you can see there's those gaps that we were mentioning. And then there's another cell that we'll talk about in a minute, the pericyte. So I mentioned that earlier. These are going to be around your continuous capillaries, and this is going to help out with helping the structure. There will be some like cartilage on the outside that will help with keeping the structure kind of intact. So your pericytes do a good job at keeping the capillary structure sound. Like I said, it's found in the skin, and with the muscle, it's the most abundant. There will be a basement membrane around this between the endothelial layer and the pericyte. And so you can see that here. You can see the intercellular junction with the tight junctions there. What is this structure? It's kind of weird. It's thin.
Is that a lumen?
A what? Red blood cell. So what helps the red blood cell be able to bend and without like breaking? Spectrin, right? We talked about that in the blood lecture. So spectrin is going to help with the red blood cell's flexibility as it's going through these capillaries. Another thing that we can mention is these like tiny little bubbles that are at the end here. This is a representation of the endocytosing and exocytosing of the nutrients that will be dispersed through the like outside of the blood and going into the tissue that's there. That's not going to necessarily be labeled on a TBL exam or anything like that. It's just to give you an idea of how this blood communicates with the tissue surrounding with that nutrients and oxygen and stuff like that. Here's another good picture. So you can see all those red blood cells that are like lined up in the endothelial layer that's lining. So this is like a longitudinal section. Right? Here's a good depiction of these pericytes. So this is an EM. You're seeing this is the cell body of the pericyte, and then the cytoplasm will go up and stretch around these capillaries to help with this structure. So this will be the continuous capillaries, right? So this is definitely going to be one that could definitely be like pointed on and then say label this, like what is this cell? For sure. Next we're talking about fenestrated capillaries. So these contain the pores that I was mentioning, right? They're covered by this membrane, all right, but they're still more permeable than the continuous capillaries because of those pores. This found in the body where a lot of absorption and filtration takes place. So, like the kidney, the intestines, endocrine glands, you're going to have like your thyroid, parathyroid, stuff like that. That's where you're going to find the fenestrated capillaries. So in the kidney, there will be filtration. Intestines will be absorption, right? And then endocrine glands, you'll have your hormones being able to communicate to the endothelial cells. So it asks, where can you find fenestrated capillaries? Any of these three would be okay, right? And then here's a depiction of that. So still has tight junctions, but because of these pores, it's allowed to have more nutrients flow through, right? And that's good for the filtration and the absorption. So you can tell the difference between this type of capillary and the continuous one that we talked about, right? These pores, and we'll actually be able to see that in the picture here. So here you can see that they call fenestrae. There's these little gaps within the endothelial layer, and those are the pores that we're talking about. This diaphragm was that basically this membrane that goes around, right? That's covering and helping protect. And then you can see the endothelial cell that's surrounding the end with this red blood cell that's going through. Last one is sinusoid capillaries, right? There's fewer tight junctions. They have larger intercellular clefts, so they have big spaces. These allow for large molecules, some even cells, like I said before, to be able to pass through and communicate. Only found in the liver, bone marrow, spleen. This last one is questionable, but in terms of our class, it'll work. The adrenal medulla, some people say it's fenestrated, some people say they're sinusoid capillaries. Right now, we're just going to go with the adrenal medulla being sinusoid capillaries. So again, questions on, where can we find sinusoid capillaries? Liver, bone marrow, spleen, and adrenal medulla. And then here's that depiction enlarged again with sinusoid capillaries. So you can see, compared to the pores, these are a lot bigger structures that are allowing that nutrients to flow in and out. Right? You see the endothelial layer, but it's broken up by those spaces. There are some tight junctions still, but they're not as in abundance. This is the most permeable type of capillary, right? So permeable meaning that nutrients and oxygen and stuff like that is going to be able to flow back and forth. And again, with these capillary beds and network, right? So we're going to talk about the structures that I was talking about with the sphincters and how blood goes from the arterial to a metarterial and then to venual and throughout, right? So it goes from the arterial to the venual through this capillary bed. Contains these two types of vessels. There are a couple other things that are called a vascular shunt, which is just, I'll show you here in a second. This right here. So this vascular shunt is going to be this region between the metarterial through what they call a thoroughfare channel. And we'll see that here in a second. The thoroughfare channel is where you're going from this arterial, the metarterial, and then all the way through the center. Do you see how there's no sphincters on this one here? Compared to if you were to go any other route that's there, you have these sphincters of smooth muscle. that are able to close off these capillaries. And these capillaries that are in that region, from where the sphincters are able to flow through, are called the true capillaries. So this is where you're going to be able to see, like for this instance, would be like the continuous capillaries, the fenestrated, the sinusoidal capillaries will be down here in your true capillaries. And this thoroughfare channel is going to be this blood that's flowing all the way through, even if these sphincters are closed. Anybody guess why that blood will flow through, even if the sphincters are closed? Why that would need to happen?
Pressure.
Maintaining
Yeah,
pressure
junction.
could be one, yeah.
Why the sphincters close?
Like if the sphincters are closed, why does blood still flow through this channel? Just through that this little channel here that isn't enclosed by the sphincters.
There's a lot of driving force behind it. It just
Yeah, so-
it's an open door
Yeah,
basically.
so it's an open door, there's a lot of pressure. Functionally, that tissue still needs to stay alive, right? So we can't completely shut off the entire tissue from blood. So when I was mentioning when you're running away from the bear or something, you're shunting that blood from your muscles, but there still is a little bit that's going through this channel that's keeping the tissue alive.
Okay. So the capillary bed that could be like for I don't know, like your intestines or something, like your whatever you don't need to
Yeah.
run away from a bear.
Yep.
Gotcha.
And so that's what these sphincters do. These sphincters are regulated by the autonomic nervous system as well, with the arterioles as well, along with that. And so with these will shun off these true capillaries, just allow a little bit of blood flow to go through. And then if you need a lot of blood flow to go through, sphincters will dilate, and then that blood will flow through the true capillaries down at the bottom and on the top here, right? And then they'll go in through these venules and then out the vein. Right? Let me go back here and we'll see these definitions. So this vascular shunt is this area that is from the metarterial to this thoroughfare channel. So it's the part where that channel that I was talking through, where it's continuous blood flow, whether the sphincters are closed or not. That would be your vascular shunt. It's also called intravenous anastomosis, and I'll start there. Anastomosis, there are a couple different terms you can either write down or just know to understand them, because we'll talk about them a lot in growths, and we might need to know on a question with the exam or something. So just to clear it up, anastomosis is the union of the branches of two or more arteries supplying the same region. Okay? And then with that, there's collateral circulation. Collateral circulation means an alternative pathway for blood to flow. So in neuro, you learned about the circle of Willis. So that would be an example of anastomosis and collateral circulation. So there is arteries going to the same region of similar tissue, and then if one is closed off compared to the other, there will be a collateral circulation, meaning that the blood has an alternate route to still supply the tissue. Okay? So this vascular shunt, like I said, that's that region that directly connects the arterial to the venule, so bypasses the true capillaries if the sphincters are closed. The metarterial is labeled, right? So this is the intermediate between the arterial and the capillary. It's continuous with the thoroughfare channel, so as it's coming through, it's going through that channel. And then this is also surrounded by the smooth muscle cells that have the sphincters, right? Which we'll label here in a second again, just to get a clear picture. And then we already mentioned the thoroughfare channel, right? So it's the intermediate between the capillary and the venule. So these are just labeling the regions within this capillary network. So here we go again. So this is our, our capillary network. We have our arterial. This structure right here is called the metarterial, right? So this is going to be continuous with your thoroughfare channel, which is this label here, right? So if the sphincters are closed off, blood will flow through the metarterial thoroughfare channel and then down to the venule. If they're open, then it's going to go through the metarterial, down through these true capillaries, and then up to the thoroughfare channel here, and then to the venule. Does that make sense? All right, true capillaries, which I mentioned, so those are these down here. They branch off of the metarterial, and they can be shunted due to those sphincters, right? So this is where you're going to have the most exchange between the cells in the blood. So when you're wanting a lot of nutrients and blood exchange in the certain tissue, those sphincters are going to be open. So this is going to allow it to go to the true capillaries, which handle most of that nutrients exchange. All right, so that's there again. So the precapillary sphincters is what I was talking about, the sphincters, right? So those are the ones that will close off and open up depending on what type of blood flow we're going to need in that area at the time. These are made up of smooth muscle fibers, and they're on each true capillary section from the metarterial, right? So when we're looking here, there's going to be a precapillary sphincter at each part that goes into the true capillary. And then these will branch off to then match up with these, the thoroughfare channel, and then under the venule. And so in these regions, that's where most of the blood exchange is going to occur, through these capillaries. I mentioned this, and you're just seeing this again. When the precapillary sphincters are relaxed, that means they're dilated, right? Blood flows through them and through the true capillaries. If they're contracted, blood shunted, and it goes through that vascular shunt, which bypasses those tissues and just goes through the thoroughfare channel and then into the venule, right? And like I said, the reason that it does that is so that the tissue doesn't completely die off. It gives it a little bit of oxygen so that the tissue can be stable enough to survive. And then it's regulated by the autonomic nervous system, right? So this will be your sympathetic, parasympathetic. So here's a good depiction. This is saying that the sphincters are relaxed, right? They're open. Blood's going to the true capillaries, and then it's going up through. The blood is being flowed out into the tissue. All the nutrients is leaving. So this is where all the gas exchange occurs, and then it goes through the thoroughfare channel into the venule and back out. If these are closed or closed off or shunted, it's going to go through the metarterial, through the thoroughfare channel, and the venule, and that's it. It's not going to go through these true capillaries. All right, so this is a good picture to kind of know the difference between when the precapillary sphincters are open and closed. Why are these sphincters important? I already mentioned one about shunting blood to different parts of your body, right? But it also helps with body temperature. So when your body temperature increases, sphincters in the skin will relax, allowing more blood flow to the skin to release heat. Right? So if you're overheating, blood will flow to your skin so that your skin can release that heat from sweat. Right? If your body temperature decreases, the sphincters will contract so that it will keep the blood toward your internal organs and keep your main body temperature at a higher rate. So it's important for that, right? Doing the same process that we already said. Secondly, if you eat a large meal, I mentioned this before, that you need enough blood to go to your GI tract when you're digesting stuff, right? So your parasympathetic nervous system is going to turn on, the sphincters are going to relax and dilate, and then the sphincters that are in your extremity will constrict, so that you can have most of the blood going through your GI tract to be able to digest food, be able to exchange nutrients and stuff like that. And so between meals, the sphincters contract to reduce the blood flow, after you're done with digestion and everything. And then the last example is during exercise, right? So this is what I was mentioning with the fight or flight. You're going to run away from the bear. You need a lot of the sphincters open for your skeletal muscles so that all that gas exchange is going to occur, so that you have the oxygen for your muscle, which will then create energy that you're going to need immediately, right? And then it'll also go up to your brain so that you can think critically to get out of a situation or things of that nature. So that's where the gas exchange nutrients are needed at that time. And your body does this all by the autonomic nervous system, so it does it without your control, which is interesting. So here's a nice graph to go from the very end to, or the very beginning to the very end. So you're going to start off with an artery, right? We're going to go to where? Arterial, and then what? Yeah. So arterial at the end of the capillary, which would be your met arterial, right? And then your capillaries in the middle. Then we're going to go to what?
Venules.
Yeah. So the venual end of the capillary, which would be, depending on those, you'll have your true capillaries, and then it'll go to your venule, and then venule to vein, right? So you're going from a larger structure, going down to where the pressure is not too bad for your capillary to exchange that nutrients, and then it's going back up to a larger structure to where the blood collects, and then the veins will have these valves that will be able to put that blood back up into the heart, right? All right, so we'll get through as much as we can with this heart section, and we have a little bit of lymphatic, and then you'll be ready for your lymphatic lecture next week. So the heart has 4 chambers. Anybody tell you differently, then they are incorrect. If they told you that, it's probably because they had anatomy class and their undergrad that didn't deal with cadavers, and they did it with animals, like for instance the frog. Frog has 3 chambers, right? It has 2 atrium, 1 ventricle. In terms of anatomy, we can describe, from the frog and get a lot of the same ideas and concepts, and so it works for the most part. And a lot of anatomy programs don't have the money to be able to produce a cadaver lab if they don't have like a master's program or a med school attached. So they'll explain to that. So you get some med students that will come in and they'll be like, oh, there's 3 chambers to the heart. And then they're completely wrong, obviously, because we have 4. 2 atrium, 2 ventricles, right? And that's what we're going to get into just briefly for the next 10 minutes or so. So it has 4 chambers, right? We already talked about the type of muscle that's within it, the cardiac muscle that it'll have, right? It has this fibrous central region called the cardiac skeleton. It's going to be formed by dense connective tissue and I believe fibroblasts as well. So you wouldn't think that it would have those. No, not fibroblasts, fibrocartilage. Made a note on it. Just want to make sure I'm saying everything. Yeah. So this cardiac skeleton will have this dense connective tissue and some fibrocartilage, and that's going to help support the outer portion of the heart. Serves at the base of the valves for the attachment and the attachment of the cardiac muscles. So it's kind of weird when you're thinking about skeletal muscle, right? You have the bone for the muscle to attach to. Your cardiac muscle, you don't really have that. So what does it attach to? It attaches to this dense connective tissue and fibrocartilage, so that your muscle has something to grab onto, so that when it contracts, it actually creates a movement, right? We have these cardiac valves, right? So again, with that dense fibrous connective tissue, and they have the endothelial layer, right? Because blood's going through the heart, so you're going to have a simple squamous endothelium. And then again, like I said, their bases are attached to these annular rings, the cardiac skeleton that has that dense connective tissue and fibrocartilage. Layers from the very outside of the heart, as we get closer into the tissue. We talk about viscera in gross anatomy. So your lungs have viscera, your heart has viscera, right? There's some different structures in your gut that we'll talk about later in the semester. So your heart is surrounded by this double-layered sac, and it's called a pericardium. If you think about this in terms of how it looks, like it's kind of hard to do it without dissection. But a good way to describe it is if you have a balloon, and I blow up the balloon, and I stick my fist in it, that balloon's gonna wrap around my hand, right? That's like your pericardium. And instead of air being filled in that balloon, there's gonna be this fluid that we'll talk about that helps when your heart contracts and that balloon surrounded on it, it helps with friction so that there's less friction because you don't want a lot of heat generated from your heart pumping. And so this fluid cavity that is created by the pericardium allows your heart to pump with not as much friction so that everything can be nice and happy. This pericardium is composed of fibers, and then there's also the serous pericardium, which will have a different name for it, so I'll tell you that in a minute. The fibrous pericardium, it's a superficial layer, it's that balloon that's surrounding the heart, right? It's composed of this connective tissue that we mentioned. Serous pericardium consists of parietal and visceral pericardium. So your fibrous pericardium is on the very outside, and then on the inside of that fibrous pericardium, you're going to have this parietal pericardium, and then the viscera means what? Organ, right? You mentioned like the GBE and stuff. Visceral means organ, so that's going to be the part that's actually on the organ, and we'll have another name for the visceral pericardium, which is going to be epicardium. So these names are interchangeable, the epicardium and the visceral pericardium. Your parietal pericardium is on the inner layer of the fibrous pericardium, right? So it lines the inner surface. And then the visceral is going to be on the actual organ, right? So it's going to be the external layer of the heart. So when you dissect everything, when you take off this fibrous pericardium, you're going to have this layer that should be filled with fluid, right? In the cadavers, you'll see some fluid, but you won't see it like it is in like the living tissue. And then the direct layer on top of the heart is going to be your visceral pericardium. And so, like I said, visceral is organ. You can also think of epi on top when we're talking about like the muscle and nerves and everything. The outer layer of that organ is going to be your epicardium or your epimysium, epineurium, right? So those two things will help you remember the difference between these two. And then the pericardial cavity is just the cavity that is between those two, the parietal and the visceral pericardium, and this is where that fluid space is going to be containing, right? And I said that was the help with the friction of the heart's contraction. And so this is just labeling the fluid within that cavity. So here's a depiction of it. So you have the pericardium. So it's this outer line, it's kind of like lined in white. And then the blue is going to be your parietal pericardium. And then directly on the heart, right, the tissue that's surrounding all there is going to be your visceral pericardium or epicardium. So your pericardium is broken up into two sections, like I said, fibrous and serous. Fibrous is going to be the very outer layer. Serous is going to be the layer that contains the parietal pericardium, the pericardial cavity. So you'll have the parietal layer that's on the inside of the fibrous pericardium in the blue, and then directly on the tissue will be the visceral. Questions on that? All right, so like I mentioned, with the heart, once you get to this epicardium, or the visceral pericardium, it has the inner layers that we'll talk about, and we'll see it like a little chunk that's pulled out of the heart, and we'll see all the different layers on them. So these three layers are epicardium, myocardium, and endocardium. Anybody guess what myocardium has?
Muscle.
Muscle, right? Myo, meaning translation is muscle. This is where all the cardiac muscle tissue is going to be at. And then the endocardium is going to be where the endothelial tissue can be at, right? So epicardium, we already labeled, we know what it is, right? Myocardium, cardiac muscle, we said that. Endocardium, this is going to be your epithelial tissue, specifically simple squamous endothelium, right? And this lines the internal surface of the heart. When we look at this picture here, you can see this structure, they call it trabecular carne. What's a little different with the heart than what you would expect is it's not a smooth layer on the inside. It has all of these trabecular carne, which you'll get into more of what they do with growth. But this simple squamous endothelium, this endothelial layer, is going to be that layer right on top of this trabecular carne. Okay, so it's going to be your first layer. that's inside. So this would be the inside of the heart. This is the outside of the heart up here. So then this middle layer is going to be your myocardium with all that cardiac muscle tissue, right? So then if you're thinking about questions with that, it would be, is this muscle tissue striated? You know, does it have gap junctions? All those like different things that we talked about. Where's calcium stored? Things of that nature. And then the epicardium, which we talked about, or the visceral pericardium, is going to be this outer layer that has fat tissue and connective tissue. You'll see in gross next semester that when you're taking apart the heart, they're actually, depending on the heart, there will be a lot of like fat connective tissue on the outside, especially if the person lived not as healthy as others. That fat tissue can build up on the heart a lot more. This outer layer of this epicardium, right? If we remember back to the first exam, what's the layer called of simple squamous epithelium that's on the outer edge of the organ? Yep, mesothelium. So this would be simple squamous mesothelium if we were to tag on the outside of this cardiac tissue. And on the inside will be simple squamous endothelium, right? All right, so now we're going to talk about the conduction system. We're going to leave off here, so I'll briefly just touch on it, and then we'll pick back up here on Monday, right? And then we'll finish this and go back through. So very quickly, I'll label these structures, and then we're going to see just what it is, so you get an idea of where we're moving to next. This is an impulse conducting system, right? So your heart is able to stimulate its own contraction, which is super cool. The way it does that is through this SA node or sinoatrial node, right? And the atrioventricular node. And you have what they call this bundled hist for atrioventricular. They have it labeled differently here. Yeah, atrioventricular bundle. So the SA node is responsible for initiating the contraction. And what happens here, you can see, remember, heart has 4 chambers in the human. So you have an atrium over here, another atrium over here. Specifically, this is your left, even though it's on your right side. Remember, a lot of the time anatomically, we're looking at things flipped. So don't get confused. This will be your left atrium. This would be your right atrium, and then your right ventricle and your left ventricle, right? So we're talking about this conduction system with these special nerve fibers that are here. Your sinoatrial node is what's going to initiate the contraction, so they call that sometimes a pacemaker, right? So if this conduction system is not working properly, that's why people have to have a pacemaker. And what it does is it just gives your heart a little bit of a stimulus so it's able to contract on its own periodically at the right intervals. So your SA node is going to make this signal. First, it's going to send its signal to the atrioventricular node, and at the same time it does that, it's going to send a signal to both atrium. And so that's why there's this like hiccup with your heart. It's because the sinoatrial node will tell the atrium to contract, and then once this atrioventricular node processes that signal, then it's going to send it down to the ventricle. So it's going to be atrium, then ventricle. And so it's this squeezing that's back and forth between the two. Here's the bundle of his. That's where this atrioventricular node will flow down, and then in each separate ways will go to each ventricles. And then there's some special cells that we'll talk about that's on the outside of this cardiac muscle tissue layer called Purkinje fibers. And so I won't go too in depth with that, but those are special cells that we look for that look like this. And so I'll go more into that on Monday. Any questions? No? All right, go take a nap and go enjoy the weekend. Don't nap too long, so you can do stuff tonight and go over it on the weekend. Cool.
Awesome. Huh? I'm gonna remake the notes and make some class cards, so that way if I have any questions I can ask him all around the the review. I was gonna go work out.
Can you believe it?
review,
They're still making
do
us
some
come to
back
class.
because I haven't working out at all.
Yeah.
And I tried to do some pull-up yesterday, and I'm like, dude, I can barely do like 6. Like, I'm definitely getting weaker.
I forgot we had that seminar. Like, I need to do my paper. Dang it.
Wait, we don't have a live seminar, do we?
What are you doing?
It's just a paper.
No, the live seminar was.
I'm gonna go live tomorrow.
Yep.
Okay, so
Everything's online.
after, I'm gonna go to the gym, go home.
Yeah.
I don't know what I'm doing after that, probably homework for a seminar.
Okay.
Finish up in an hour, and I should have done it yesterday, but I just kinda like relaxed
Oh, if you wanna
yesterday.
see your CV, I'll have
Okay.
that too.
I do.
Before you leave.
I know what I missed, I just need to see it.
Did a lot better this time, guys, good job.
What was the average?
Average was an 83.25. And we had quite a few A's.
I don't even.
I thought I knew about it, but then I wasn't like, no.
You knew more than I did.
Yeah, I
But
thought
yeah,
it
you
was
go
the
to
blood,
the-
I thought it asked the cell of the blood nerve barrier and then the structure of the blood nerve barrier, so. I misread
I've got to
it.
learn
I misread
the
it.
question.
So the questions for
No.
Yeah. Oh, yeah. Same with
I
neuro
missed
too,
out
though.
one question.
Which one?
Because the satellite cell, because I wrote where it was, but I didn't write what the cell it was. I just completely skipped over it.
Yeah, that's, I'm not good at talking about the head.
Oh, really? Yes. Oh, gosh.
I'm scared.
What?
What?
Wait, you're going in
Yeah.
person?
Yeah. I'm going right freaking now. Now I'm gonna say.
So was it the material?
What are you gonna
or
say?
because
Well, he
I don't know,
was
muscle
like,
lecture or?
gave me my grade afterwards and was like, you still wanna come in? And I said, yes. Because I still didn't know I got it.
Or is it
Really,
just
I missed.
like-
I thought I got
Did it go on the-
I think people are
Questions, right?
taking it more seriously, I think.
Yeah, I'm just playing a joke.
He
I
said
thought
that
that's
he had
what he was
3
saying.
series
More
on
on
it.
that.
And that I missed
It's all you, don't stroke your ego too hard though.
Everywhere.
I wonder why when they gave it, why they didn't like split up the extended from the.
Yeah, well,
Yeah, I know.
When you don't actually know like how many you got right.
Yeah. Yeah.
Unless, did they just throw out all the extended response?
I don't know. I just know that everybody's in an exam that was that bad that they had for that says a lot.
Yeah, that's probably.
It did help though.
I mean, yeah, but that says a lot of.
Like, I'm either here or I'm by myself.
And like obviously,
Like, whereas like
the
Alex
shit
would be
that
here.
I was
Like
like,
if
that
Andy
question
was in
that,
my
I don't know
lab,
if you heard me
you're
talk
here,
about it in the class
then
yesterday,
Yeah.
but I was like,
I
well,
have a
that's
buddy,
great
we go to
because
the lab like every day,
nobody ever
like,
told
work
us
stuff.
that there are
They
two
didn't.
synapses, like sure, we can see on the picture, but.
We didn't talk about them.
Yeah, just the explain how they have to figure that out.
But then I'm like, come on, concentric looks like iron, which what the Brazilian looks like.
Yeah.
So I was.
So I'm looking forward to seeing what I missed
I'm like,
on the
maybe
exam
it's not
response, because I
true.
feel like I did well. I don't know what I want.
I could not.
Is it
I think,
Brazilian?
you know, when it was like,
So what master
you
is
said the whole some of the wording in my brain, I was thinking like, yeah, I miss all of Dr. K stuff, but I couldn't just say that.
Yeah.
Like the
The wording.
wording.
Well, it was though.
It really is, like,
See you, man.
and I'm looking forward to seeing too, because on, I think a couple of the questions, he was like, ultimately, what happens? And I'm like, well, ultimately, means like ultimately.